Hey guys! So this is going to be our first online video recording slash lecture. Sorry that this took a little bit longer than expected to get this out to you guys. I know I posted this slideshow last week, but I had some technical difficulties. Turns out that my laptop isn't so good at recording audio. So now I have a nice microphone. We're all good. Hopefully you guys can hear me okay. Alright, so this lecture is going to be on chapter 14, species and speciation. Hopefully you guys have done this homework. If not, you still have time to go back and do that or redo it. But I'm pretty sure that most of you guys already have, so good job on you staying on top of everything. So species and speciation. We've been talking a lot about species and speciation already this semester. But now we're going to actually spend a whole entire chapter looking at just what is a species and how does speciation actually happen. Alright, so this is one of the most debated topics in evolutionary biology. It has been debated since Darwin, even before Darwin wrote his book. And some things that we ask, especially when looking at species or speciation, is what even is a species? Like, how do you draw this arbitrary line between two organisms and say this is a species here and this is a species here? How does it actually happen? Can we document it happening? Can it happen quick? Does it happen slow? Things like that. What creates reproductive isolation among populations? So we'll look a little bit more into that, what reproductive isolation is. And what do evolutionary biologists know about the genetics of speciation? And so that'll be at the very end, and we'll just spend a minute looking at that. Alright, so the species problem. So Darwin's book was named On the Origin of the Species, and this title was not chosen just because it sounded good, but it was chosen very, very carefully, because the whole reason Darwin developed his theory of evolution by natural selection was to literally try to understand and or explain the species problem, or how can we account, or how can we explain all of the different life forms that we see on the planet today, and how could it have gotten to where it is? So like looking at all of the past species all the way up until how everything looks the way they do today. Okay, and also, what is a species and how do I identify species and delineate these species boundaries in nature, right? So we can obviously say like, okay, this species of cat is obviously different than this species of dog, but when you look at different types of cats, how do we go about actually identifying which are species and how do we draw that line? Okay, so there's a couple different models or concepts that attempt to explain these things. And so the very first one that we look at is the evolutionary species concept. And so basically, this is the idea that when a group of organism is a species, that whole group or species forms a lineage on like a phylogenetic tree that has a distinct evolutionary fate than that of other lineages. So like that group of species will go on to become like 3 new species or something like that. Unfortunately, the drawback to this one is it's more conceptual. So like if you're imagining like a phylogenetic tree with those like branchings, like think of like a node on that tree, and how we say that that node must have been maybe a species and it gave rise to a multitude of new ones. But what this doesn't tell us is that It doesn't give us practical advice, or it doesn't give us an actual way of separating or creating species boundaries of things that are alive necessarily, like today. And so there are 4 other concepts that we use to do this. So as we know, we can get a lot of weird hybrids. They're not necessarily the norm. So we know that there are tigers in nature. We know that there are lions in nature. And generally, they do not overlap like where they actually occur. For the most part, tigers are in Asia. Lions are in Africa. But when we have them in captivity together, I wonder if any of you guys have been watching Lion King. Or not Lion King, Tiger King. Yikes. Anyway, but we get this hybrid called a liger or a tigon. And a liger, I think like the tiger is the mom, and with a tigon, the lion's the mom. It doesn't matter. But so, right, we have lots and lots of tigers in nature. We have lots and lots of lions in nature, but we really don't get these guys. Another thing that's kind of similar to this are like we have lots of horses, we have lots of donkeys, but we don't necessarily have a lot of mules. So we have these 2 species. They look pretty different. We have these stripes, orange, white, black for the tiger, whereas with lions, the males have a mane. Tigers typically don't have manes. But they look very, very different, right? So that is our first species concept for things that are alive today, or being able to draw lines between species. And so organisms are typically clustered together in phenotypic space, meaning that we have a lot of things that look like this. We have a lot of things that look like this. Not a lot that look like that. So we have these clusters here. Whereas like that liger, that tigon, we might have scattered somewhere in this plot, not in these clear-cut like boundaries or lines. And so this concept is very, very commonly used by like numerical taxonomists or people that really like numbers, people that like doing statistics. And so you can look at these 3 species up here and be like, okay, well, they kind of look alike, but you can measure different things, measure different traits or patterns seen in these species, and you can plot them like you would anything else, and you'll get things that cluster together just like this. There's a couple issues with this though, and you might have already been thinking this. What traits or characteristics should we use? Like, so for our cat example, should we use size? Because would that tell us like size of the animal, weight of the animal? Should we use like gestation time? Should we use color? What should we use? And so that's one of the issues. And then another thing that you might have already been thinking already is convergent evolution, right? So maybe these 2 organisms really don't share a evolutionary history, but maybe by chance they have similar colors or they have similar shapes, but perhaps this wasn't caused because they share an evolutionary history. It could be because of convergent evolution. And with that being said, this idea is still used a lot today. They do use it a lot for things like fossil remains, plant and microorganisms. We typically don't use this a lot for animals. Alright, moving on. So next we have the biological species concept, and you may or may not have heard of this before, but essentially what the biological species concept is if 2 individuals can successfully mate with each other and produce viable offspring, then they are a part of the same species. Now, this could be tricky, right? Because perhaps two individuals could mate with each other if they were in the same area, but that doesn't necessarily matter, because if they are not actively mating with each other, then for the biological species concept, we do not consider them the same species. And so patterns of gene flow is what determines species boundaries in the biological species concept. Okay, so again, if species or individuals in a species can mate with each other, they are considered the same species. And so going back to our tiger and lion example, so what about these weird hybrids like a mule or a liger? And I'm sure there's many others that I'm not thinking of right now. But so what we say is they consistently mate with each other, that they're all, like, in nature, they're always going to mate with each other. And also very important, and you've probably heard this many times, but the offspring must be viable, so that the offspring of a mating, those offspring must then be able to go on and also mate and have viable offspring. So some issues with this one is that there is practical difficulty, meaning that this one is hard to apply to extinct species, because how can we say whether or not extinct species were actively mating with each other and there was gene flow occurring? Sometimes, right, we can infer this. If we have one species that lived in North America versus one that lived in Australia, even though they might look very similar, we can probably say that they were not mating with each other because they weren't even on the same continent. Another issue with the biological species concept, and the most severe limitation, is the fact that it's limited to only sexually reproducing species. And we're going to get into this a lot on our next video lecture. It's all going to be all about sexual and sexual reproduction. But so the biological species concept isn't able to be applied to asexually reproducing species, because in this case, something like a starfish or a hydra, something that only, or most of the time, only asexually reproduces, well, it's not mating with anyone or any other organism. So are we going to say that it's not a species? And that's not the case. So that's one of the most severe limitations there. Alright, next we have the ecological species concept. And so a species is considered a species when they live or occupy the same niche or niche. So what we say is if two lineages have evolved adaptations allowing them to occupy two different niches, then they are different species. And so a lot of times, and I actually have first-hand experience with this one, especially with invertebrates, so freshwater mussels or insects, a lot of times they'll look very similar to each other. They might have similar genomes or genes, but then when we actually look at their niches, like where they're found, they're in completely different areas. And so they are probably not mating with each other. And so that is one of the biggest problems, unfortunately, with this concept, is that it's actually very hard to say what the niche is of a species. So identifying the realized niche, so where they're actually found, is very difficult in reality. Because, for example, we have fundamental versus realized niche. So fundamental is where, in theory, they could be found, but realized niche is where they're actually found. And so in this case right here, we have two ecological niche models of an ant species, and so where it's very dark blue is, and the green is the fundamental niche of these ant species, okay? But if we went out there and we went to all of these areas that are blue, we probably wouldn't find these ant species in every single place. And then we might also find them, we could technically find them out in the yellow. That's less likely, but we definitely could. So we run into some issues here, because maybe this species on the left here, maybe it really is only found in like this eastern part of Texas, and maybe this one's also only found in the eastern part of Texas. So we could run into some issues here with that. But so going back to the freshwater mussels is, so when you have two species that look very, very similar, might have similar genes, and again, if you map out at least their fundamental niche, you can get an idea of whether or not they might have some niche overlap or a similar niche, and that might help you parse out whether or not maybe they are or aren't actually a species based on this concept. Alright, lastly, we have the phylogenetic species concept. So this is looking at character differences to distinguish among species. So this is kind of similar to the first one, looking sort of at clusters. But so we draw these species boundaries using shared derived characters. And right, we have to be very careful about this one, because how can we be sure that characters are derived? So we have to have a big picture of a lot of different species that we may be looking at, not just like one or two. So all of these concepts, all of these models are pretty good at explaining how we potentially draw species lines or boundaries. And obviously, no one concept will work for all organisms. Each one takes a slightly different way of going about it. But for the most part, one or two or all of them usually give us a pretty good idea of where we can draw or create like species boundaries. And usually they all line up pretty good too. So usually populations that belong to different species, they show these very large phenotypic differences, like we saw with a tiger and a lion. They look very different from each other. There is no gene flow, they're not mating, they live in different areas, and usually these populations have shared derived traits. Okay, so now we're going to talk a little bit about modes of speciation. So how do species become species? And how did such a diversity of different species come to be? And you may remember that we actually read a paper about this. I think this was the first paper that we read about models of speciation. So we have allopatric, parapatric, and sympatric, and you guys probably remember these. You might not remember which one is, but we're going to go over that in a second. And these models are distinguished from one another based on geographic positions of the populations undergoing speciation. Okay, so here we have allopatric speciation, and you can see that there's two different types grouped within allopatric. And for the most part, allopatric is assuming that there is a geographical barrier to dispersal. So once this happens, they can no longer mate with each other. We have parapatric and sympatric speciation, and generally there is no geographic barrier in these cases. Okay, so allopatric speciation, the central premise here is that reproductive isolating mechanisms evolve, or two populations become separate species while they are geographically isolated from each other. And so even if for some reason that barrier went away in the future, they would still be reproductively isolated and would not be able to mate with each other after it went away. And so some good examples of physical barriers can be things like mountain ranges, rivers, oceans, deserts, etc. And so you can see, or maybe imagine, that these physical barriers, they aren't things that necessarily pop up overnight, right? These could be things that take very, very, very long time to develop. With it being said, especially after humans, we actually can create physical barriers ourselves, especially if we're talking about like smaller animals like insects or invertebrates, or if we create a dam and a river, we might create a physical barrier there. I think you guys probably understand what I'm talking about. But again, in all allopatric speciation, genetic drift and natural selection cause these populations to diverge from one another. So it could be that on this side of the river it's hotter or colder or less precipitation or whatever. So natural selection could just cause these two to diverge. But it could be genetic drift. And as we remember, genetic drift is completely random. And so because they are separated, certain traits might be more common here, less common here, etc. And so, as I mentioned, allopatric speciation can be divided into two, like, subtypes. So we have vicariance and peripheral. So vicariance is when populations are both still relatively large or both the same size. And in this case, it's most likely going to be natural selection that causes these populations to diverge, because as we remember, the smaller the population is, the more susceptible it is to genetic drift. And so that's what we see in peripheral allopatric speciation. So one population remains very, very large, where the other or multiple other populations are going to be a lot smaller and therefore more susceptible to genetic drift. And so a lot of times we could see this in a mainland and surrounding islands around that mainland. And so here, again, in vicariance, we have a geographic barrier and both populations remain the same size. Whereas in peripheral, we have pretty much the same size original population, and then a smaller population. And then it's not drawn here, but we can assume that there is some type of physical barrier here. Alright, now we're going to start talking about parapatric speciation. And so the next two, we can assume that there are no geographic barriers present. And so parapatric speciation occurs when two adjacent populations diverge into separate species in the absence of a geographic barrier like we saw in allopatric speciation. So here we have a species form here, and we have this hybrid zone, and over time we see a much smaller hybrid zone. The core concept with this one is that there must be some type of Klein or spatial gradient in the frequency of phenotypes or genotypes. And so here might be colder on this side of the area, whereas this might be hotter, or this might be shadier and sunnier, whatever you want to imagine. And maybe the organisms of the population, or the individuals of the population on this side, become better acclimated to being able to survive in colder or shadier climates, whereas on this side they become more adapted to sunny, hot environments, and eventually we get this split. And again, oftentimes in parapatric speciation, a hybrid zone will exist where there is still some matings happening between the two. And usually these hybrids face a selective disadvantage because they're not necessarily well suited to living on either side of the decline. So they're not necessarily really good at surviving colder, shadier environments or hotter ones. They're just kind of weirdly in the middle, and they're not good in either one. And so if these hybrids face a selective disadvantage, that's why we eventually see this reduction of the hybridization zone, and the zone will eventually disappear or get a lot smaller, and we have a more clear cut two groups of species, or two different species. With that being said, though, not all parapatric models assume that hybrids are at a disadvantage. Sometimes hybrids could have a superior fitness to non-hybrids. So maybe they're really, really good at surviving either environment and thus have a advantage over the non-hybrids. And so that could technically happen. Another thing with parapatric speciation is that we will get what we call a ring species. So here, all of these individuals, or all these different populations, could be the same species, at least for now. And so we'll see that up here there's a lot more gene flow going on. So these probably, especially phenotypically, all probably look very similar to each other. And then population 2 has gene flow with population 5, which has gene flow with 6, which has gene flow with 7, which has gene flow with 8, and same thing on the other side. But so what might happen is we might see population 8 and population 12, and they might look very, very, very different from each other. And so we might assume that there is absolutely no gene flow between them. But then when we look at the bigger picture here, we see that there actually is technically some gene flow in some sort of way. And so here is actually a real life example that we see of these salamanders in California. And it looks very, very similar to this here. But here, it's not a lake, it is a mountain range. And so we have species that you can see that there's like gradation between some of the colors. And so they are interbreeding with each other, but then like ones down here obviously are not breeding with these ones up here. And as you can see, especially from these pictures, that these phenotypically look very, very not similar. And so technically these are all considered the same species, but they're subspecies. Alright, so lastly, we have sympatric speciation, and again, this occurs when there's no geographic barrier that's separating the species, and there's also not a cline or gradation like we see in parapatric speciation. So it's very hard for scientists to understand, like, how the heck does this happen? And so going the wrong way. So going back here, speciation happens like just right in the middle of the original population. And so the reason that they think this probably happens is due to habitat or ecological speciation. And so what I mean by that is that perhaps there is a lot of competition for a particular source of food or source of sunlight or something like that. And so individuals of the original population, some of them might be more equipped to handle a different habitat, and therefore they might speciate. And so that is actually something to what we see with this fish found in South America. And so Um, this fish is orange fish here, Amphilophus citronellus. So I guess it's named that because it looks like an orange. I'm not sure. But so this species is found in Lake Nicaragua. And there's lots of little lakes that surround it. And so we still find this species in the smaller ones. But then I believe it is Lake Apoyo here. We also find this Amphilophus zaliosus. And our scientists were confused, like, well, how did this happen? Because they do, genetically, they're related to each other, but physically, they look similar. Physically, they don't look similar. They have different body types. Sorry. And they also specialize in different types of food. And so what they hypothesized and is that there was competition between these species at the bottom of the lake. And so certain individuals may have been better at being open water foragers. Maybe they were better swimmers, etc. And they eventually speciated and became a different species so that they weren't directly competing with the original Amphilophus citronellus. And we see this a lot with plants, because we know that plants can't just like walk away. And so we see this a lot of times with different plants, trees, things like that. But we can also see it in vertebrates. Alrighty, so lastly, we're going to talk about reproductive isolating mechanisms. Scientists are very interested in how this happens. And there are multiple types of mechanisms. So we have pre-zygotic and post-zygotic. So basically before mating and after mating. And it's pretty straightforward, but we'll see, as everything for the most part that we've covered this semester, that it's not necessarily always as straightforward as it seems. So here we list out pre-zygotic isolating mechanisms. And once I talk about these, it'll make more sense. But so the first one here is mates live in the same place, but they don't encounter each other. And that could be because they come out different times of day or slightly different times of year. And so they literally just do not come into contact with each other. So if they do not come into contact with each other, well, then obviously they are not going to mate. Potential mates then could interact but do not mate. And so this could be because of a multitude of things, but maybe the female just isn't picking up what the male is laying down. But it just doesn't happen. So, right, if they don't mate, well, they don't mate. They do mate, but the male gametes are not transferred for whatever reason. And then lastly, the male gametes are transferred, but the egg is not actually fertilized. So the gametes just are incompatible for each other. And we'll talk about that a little bit more with genetics at the very end. Okay, so these are all before, I guess, a zygote ever forms. Not necessarily before mating, but before a zygote is formed. Then we have post-zygotic isolating mechanisms. So a zygote is formed, and maybe even an animal or individual. So post-zygotic isolating mechanisms is that the zygote just literally does not survive for whatever reasons. The first generation hybrids are not viable. So they just, maybe they're born, but they don't survive very long. Perhaps the hybrids do survive, but they're sterile. So like as we see with mules, we know that a mule can't mate with a mule to create another mule. So a mule is not considered a species, right? And then lastly, perhaps the first generation, maybe they're able to mate, but then their offspring are inviable or sterile. This is one of my favorite topics here. One reproductive isolating mechanism is isolation through pollinators. There is a very, very tight linkage between the feeding morphology of animal pollinators, such as like moths, butterflies, bees, bats, what have you, and then the plant traits associated with pollination. And so we know that usually the animal pollinator will want something from the plant, whether it's nectar or pollen. And so you can see in this case here with this flower this is probably either the pollen tube or the way that it's the style or stamen. I'm not actually really sure with this particular flower what this is, but anyway, whatever it is, we can assume based on this flower morphology that it's going to have an animal pollinator with probably a very long proboscis. And so proboscis is just a fancy word of saying like insect tongue. And so what's interesting about this story is that Darwin saw these flowers when he was on his journeys on the Beagle, and he predicted that there must be a pollinator that has a very, very long tongue. And he actually was correct. He never realized he was correct, but later on they found that this moth here is actually the pollinator of this plant species. And so over time, the effects of pollinators on reproductive isolation in plants, this has been documented for many plant species. I'm going to come back to this link here in 1 second. But typically, based on flower morphology, we can predict what type of animal pollinator is the one pollinating it. So in this case here, with this flower, we have these hawk moths. Whereas with B, it's primarily hummingbirds. And then A here is mostly bees. So it's very interesting how we can predict pretty accurately the pollinator of flowers based on their flower morphology. Alright, so real quick, let's switch gears here. Alright, here's a quick video.
This buzzing is a secret password, the key to a lock. What this bumblebee is after is pollen. Bumblebees eat pollen. It's high in protein. But the flower doesn't want to give it to just anyone, so it hides it away in those bright yellow anthers. For a flower, that's unusual. Most flowers keep their pollen on the outside of the anther, which is the male part of the flower. Pollen is basically sperm for plants. Most flowers make sugary nectar too. They use it as bait to attract bees and other pollinators, which get coated in pollen along the way. And since bees are messy, they inadvertently scatter some of that pollen onto the female part of the next flower they visit. That's how most flowers have sex. But this type of flower doesn't offer nectar. The only way to get to its pollen is through those tiny pores at the ends. But the bumblebee knows just what to do. It wraps its legs around the flower and bites down on the anthers, that male part of the flower. See those wings shaking? Normally, the bumblebee uses those powerful muscles to flap its wings. That's what makes the buzzing sound when they fly. But here, those muscles vibrate its whole body, so hard and fast that it makes a louder, higher-pitched buzz. This vibration shakes up the pollen trapped inside the anthers until it spews out all over the bumblebee. It's called buzz pollination, and you don't need a bumblebee to do it. A tuning fork will do. The bumblebee grooms the pollen down into sticky sacs on its legs, carries it back to the hive. Only a few types of pollinators, like bumblebees, are capable of buzz pollination. Honeybees can't do it. This field is kind of a free-for-all. Think Las Vegas buffet. Tons of food, but long lines. Lots of competition. Buzz pollination is more like a private club. By only permitting pollinators that know the secret knock, the flower ups the chances that its pollen will end up on flowers from the same club, the same species. The bumblebee? Well, sure, it has to work a little harder, and there's no sweet nectar. But it's a reliable pollen stash that almost no one else has access to. Tomatoes, potatoes, blueberries. All of these need buzz pollination to reproduce. Much of the food we eat owes its existence to that buzz. Our secret password here at Deep Look?
All right. So that was a pretty extreme example of a reproductive isolating mechanism. So I thought that was a really cool example, and I wanted to show that with you guys. All right, so lastly, the genetics of reproductive isolation. There's two main genetic mechanisms that can cause reproductive isolation, and these are changes in chromosome number and chromosome rearrangement. So changes in chromosome number, this is very common in plants. This especially happens in plants, like they might double or quadruple the amount of chromosomes they have. But when they do that, they are unable to reproduce with individuals that had the original number of chromosomes and create viable offspring. And mostly we think that this is very common in plants because plants do have the ability to self-fertilize for a little while at least, and because they're able to self-fertilize, they can keep that population going until there's more and they're able to reproduce with other individuals. And then lastly, the chromosome rearrangement, and so that is when maybe a bit of a chromosome or 2 bits of 2 different chromosomes might break off and switch. And by this happening, this individual that this might have happened to is unable to reproduce with an individual that had the original chromosome rearrangement. Okay? That is all that I have for you for this lecture. It's about 45 minutes or so. And I am going to actually go ahead and I'm going to record the next lecture as well, so I can get that one up for you too. And again, that's going to be sex and sexual reproduction. And then after that, we're going to have evolution of sociality and coevolution. So we have 3 really, really interesting, cool topics coming up, and I'm really excited to talk about them with you. As always, if you guys have any questions about anything, please do not hesitate to email me. You guys have been really doing great with this whole COVID-19 crisis. I know it's really, really hard not to be in class all together and having me like remind you guys about things, but I'm really, really proud of you guys for sticking it out and doing a really great job. Alright, I'm done being corny. I'll see you guys next lecture. Alright, see you guys later.